Good afternoon. We're going to do a Camtasia recording of the PowerPoint for topic 3, which deals with cell structure and function. You want to be aware that there's a second Camtasia recording and a second PowerPoint specifically dealing with the organelles of the cells, and there's a homework assignment associated with that PowerPoint as well and that Camtasia. That material in the second PowerPoint is a supplement to the quiz or the topic 3 material, and I would like you to do that outside of class so we can save some class time in order to cover this material and keep ourselves on track in getting through the material toward the end of the class. So here we go. Pansy's here, by the way. I didn't mention that, but she already has a comfortable place on the floor lying down, and she's pretty excited about this new topic. So cellular organization. First of all, the animal cells have a plasma membrane. Sometimes it's called a cell membrane or a plasmalemma. Occasionally people talk about cell walls. Cell walls are found with plant cells, but not in animal cells. Typically cell walls are made out of cellulose. So the plasma membrane is essentially going to define the cell by surrounding it. And what I've done here, and I may not be perfect in it, but what I've tried to do is when we have a function, I put it in red, whereas the other information is in black. So this plasma membrane is a barrier, and it regulates exchange to and from the cell. Things that come in have to go through the membrane, obviously, and things that leave have to go through the membrane. It's semi-permeable, and what that means is that some things can go through the membrane. For example, oxygen molecules can go through the membrane, water molecules can go through the membrane, ions, which are charged particles, remember from our chemistry lecture, they can go through the membrane. But other things can't. For example, large molecules, such as large proteins, things of that nature. And the reason is actually pretty simple. It's like trying to get a grand piano through a normal sized door. Right? It won't fit. It's just too big. And the access to the membrane, as we'll see, occurs in a number of different ways. But some of those just don't allow all kinds of molecules to go through. The membrane is a phospholipid bilayer, which means that it's 2 layers of phospholipids, and it includes proteins. This phospholipid bilayer is not unlike putting 2 pieces of bread together. And the best analogy would actually be to take the ends of a loaf of bread, that I think they're sometimes referred to as the heel of the bread, and putting those together so that the part that is soft is in the middle and the harder part is on the outside. The only reason I say that is because there's kind of a thin veneer, if you will, both inside and outside, made of phosphate groups. So here we have a model. First, on the left-hand side, we see there's a phosphate group here, and then we have these fatty acids, which are a lipid, kind of lipid, and then they're arranged, as I said, sort of like 2 pieces of bread. Here we'd have the crust, and then the soft part of the bread, and then here we have the soft part of the bread and the crust. So it's a bilayer. What we find is these phosphate groups interact with water on both sides, inside and outside. And that interaction is very important. Extracellular would obviously be outside the cell. Intracellular would be inside the cell. This interaction is very important because the materials to get into the cell are in a water solution, typically, and they have to go through this membrane. And if the cell membrane didn't interact with the water, those materials would not move through. Okay, so there are going to be cell markers in the membrane, and these cell markers are proteins, and they have a couple of important things associated with them. One is that these proteins allow for the identification of the type of cell. And for example, red blood cells have proteins in their membranes that help the individual identified that these red blood cells are in fact part of them or from some other organism in some cases. There are going to be receptors in the membrane. These receptors are going to interact with transmitter molecules, neurotransmitters, things of that nature, hormones that can combine with the receptors, and usually that results in a change of behavior or activity for the cell. Okay, we have the nucleus. Now we're going to do a little bit about the nucleus and a couple of other things here. Most of the material about the nucleus is going to be found in that supplemental lecture. But the nucleus, if you translate, means little nut. And it has its own membrane. In fact, it not only has a membrane, but it's a bilayer membrane. There are really two membranes, an inner layer and an outer layer. And there is a space between them called a lumen. Typically, the nucleus of a cell is very large and relatively obvious. Nuclei were identified early on when people started examining cells using microscopes. Some cells don't have any nucleus. An example would be a red blood cell or a platelet. Other cells have a lot of nuclei. For example, skeletal muscle cells typically have many nuclei. So do white blood cells occasionally have many nuclei. And this nucleus is important functionally because it's going to control cell division when the cell divides, and it's also going to control cell activity. It's going to activate the cell or inactivate a cell as conditions require. So here we have a model of a cell. This orange colored spheroid structure in the center is the nucleus, and again, that means little nut. There are some other organelles in here. We'll talk about those, as I said, in that supplemental lecture. And you can see the membrane out here has been cut so you can actually see where it is. Okay. Inside the nucleus are going to be chromosomes. The chromosomes mostly are DNA, but they also include some protein. And the chromosomes, we're going to use the analogy that they're sort of like a book that has recipes in it, a cookbook. So the chromosomes are going to control cell activity, and they determine when a cell is going to divide, and they also are responsible for making copies of recipes. Those are the genes that tell the cell how to produce various materials, proteins and things of that nature, hormones, whatever the cell does produce. There's also a nucleolus, typically. A nucleolus is not defined by a membrane, but it is within the nucleus, and it's an important area for the production of ribosomal RNA. Ribosomal RNA is one of the three kinds of RNA that our cells manufacture, and it forms part of the ribosome. Ribosomes are essential for protein synthesis. We'll talk more about that in a while. We have the cytoplasm, sometimes called a cytosol rather. So here's kind of an unfortunate thing about biology. Much of biology developed in multiple places throughout the world, and when it was developing, there weren't ways to communicate with other scientists. A scientist would be doing research and coming up with ideas and naming things. As a consequence, it's not unusual to have multiple names for the same thing, and I apologize for that, but I'm just the messenger. The cytoplasm is going to surround the nucleus, and it's going to include the organelles inside of it. It's deep to the cell membrane, so it's sort of between the nucleus and the cell membrane, and it's semi-fluid, gel-like perhaps, I guess would be a way to think of it. It has molecules and ions in it. The proteins make it a little bit less fluid, and it has the organelles. And the organelles occur in two different groups. Organelles are usually surrounded by membranes, but not all of them. Again, you'll see more and learn more about the organelles with that supplemental lecture and Camtasia that's on Blackboard. The nucleus is an organelle. It has a membrane. The nucleolus is an organelle. It does not have a membrane. And each of these organelles has a specialized function. By the way, if we use our zoological roots and look at organelle, ELLE means ruff. Right. That Pansy is unbelievable. She never misses a trick. It means something small. So these are little organs, right? And just as our big organs have specialized function, like the heart is responsible for pumping blood, and the lungs are responsible for respiratory gas exchange and pH regulation of the blood, each of these organelles has specialized functions as well, and those are discussed in that supplemental lecture. One way of thinking of this is if you think of the organelles being bounded by membranes, it's almost like rooms in a house with specialized functions, right? So you have a kitchen where you mostly cook, and a bathroom where you might shower and use a toilet and things of that nature, and a dining room where you mostly eat, and a living room where you might watch TV and relax, and a bedroom where you sleep and things of that nature. So it's not that in the case of a house the analogy is not perfect. Obviously you could do different kinds of activities in any given room. Typically though, rooms are sort of specialized, including specialized features like for example, you don't usually find a stove in your bedroom unless you're living in a one-room house. Okay, then we have a cytoskeleton within the cytoplasm. The cytoskeleton is considered an organelle. It does not it's not defined by a membrane, and these are filaments that are connected with each other. It includes microtubules, and we're going to talk about those. And the cytoskeleton basically helps maintain the cell shape. And the cell shape turns out often to be highly modified for a particular function. I remember shape and function, morphology and function go hand in hand, and this is not any different for cells. Cells also have specialized shapes that are often related to their specialized functions. Another thing that cytoskeleton appears to be involved with is movement, that it is associated with organelles such as the cilia and flagella, which we'll talk about, and those organelles help the cell move, or if the cell is fixed, they move the material adjacent to the cell. In terms of the plasma membrane, the hydrophilic, right? So philic means to love, right? The hydrophilic ends are going to be associated with the water both inside and outside of the cell, where the hydrophobic ends are in the middle of the membrane. And if you go back to the bread analogy, the hydrophilic ends would be like the crusts on the outside of the sandwich, and the hydrophobic would be like the softer bread in the middle. As I mentioned, they love water. They interact with water, which is important because typically water surrounds the cells. Okay. The hydrophobic face inward into the middle of the membrane, and they can actually be involved in transport of material through the cell membrane, which gets to be pretty important. Okay. So again, I guess I've already used this analogy. The analogy of a sandwich with two crusts for the bread. The bread surfaces would be different in the middle as opposed to the outside of the sandwich. Okay. So here we have a model of a cell membrane. The fuchsia circles here on this side and on this side are the phosphate groups. Those are the hydrophilic. And then the little tails going toward the middle of the membrane are the hydrophobic groups. Those are the lipid materials made up or that are part of this bilayer. And then these red things represent proteins that are extending, in some cases, all the way through the membrane, here and here only part way through. So at body temperature, The cell membrane is somewhat liquid in nature, sort of like olive oil, and the proteins actually can move around within the structure, and we can even have molecules that are inside the cell becoming associated with a cell membrane. A good example would be the effect insulin has on a cell. The insulin, when it comes in contact with a cell, causes transport molecules that are inside the cell to get incorporated into the cell membrane, and those transport molecules carry glucose into the cell. Okay. So, proteins have a couple of functions. One of the things that they can act as is carrier molecules. A good analogy for a carrier molecule would be like an elevator. Let's say that you had something on one floor and you wanted to get it to another floor. The first floor where the material is could be outside the cell, and the other floor could be inside the cell. If you push this object into the elevator and go to the other floor, and then you could take the object out into the inside of the cell, or in this case a different floor in the building. And so these move molecules in and out of the cell. They tend to be very specific as to what they move. So if you have a carrier molecule for glucose, it doesn't transport other kinds of molecules as well, generally. It also, some of these proteins act as channels. And the analogy of a channel is sort of like a pore or a doorway between two rooms. And if the door is open, and by the way, these channels may be open sometimes and not at other times, if the channel is open, then the molecules or ions can go into or out of the cell through that opening. And that they also are very selective as to what can go through. Typically, the things that go through the pores of a cell or the channels of a cell are relatively small. Okay, so again, the red going to be the proteins, and we have transport going through at those sites. Here we have a carrier molecule that is taking a specific kind of material from the outside to the inside, or from the inside to the outside. And again, I've made the analogy of the freight elevator moving a piano between two floors. Here we have a pore, and we have something moving through that pore that's relatively small, typically anyway. So the pore sites are typically proteins that are within the cell membrane, and they let these ions or small molecules pass through. The things that go through, again, water molecules, oxygen molecules, things like that. Okay. So here we have water. We have sodium going through the pores. Potassium is the capital K. Chloride ions, small molecules like urea, things of that nature. Typically, the maximum diameter of a particle that can go through these pores is about 8 angstroms, or 10 to the minus 10th meters. That's a decimal point, 9 zeros, and a 1 meters. You don't have to be a brain surgeon to realize that that's a pretty small particle. Okay. The protein channels, how much of any given cell is comprised, or the cell membrane is comprised of these, varies between 1 and 10% of the cell surface. And as I mentioned, the pores are selective. They're not all the same. So there are some pores that allow one kind of particle to go through but prevent others, and then there are some that move other kinds of particles through. Okay. As I suggested, these pores aren't always open, and so this is something the cell can control, and that way it can regulate what comes in and or out and what doesn't come in or out. Excuse me. The proteins also give the cell structural support and tie the membrane to the cytoskeleton, and this makes the cytoskeleton relatively stable. Some of the proteins act as enzymes. And enzymes we're going to talk, we already talked about, I guess, enzymes a little bit. Enzymes are chemicals that alter reaction rates. We'll talk more about that in a bit here. Sometimes these proteins are receptor sites for messengers, such as neurohumors or hormones. Hormones are chemicals that can control cell activity. Either they can turn the cell on or off for certain kinds of activities. Neurohumors are very much like a neurotransmitter. These are chemicals that also can stimulate the cell to be active or to become inactive. Some of the proteins are responsible for adhesion, and so this is a way that cells can stick to connective tissue or to other cells. This probably doesn't seem like it's very important, but if you think about it, if your skin were sliding all over the place and not really attached to the underlying tissues, your body would be in a constant disarray, I guess would be a way of thinking of it. Also, some of these proteins help us recognize which cells are ours. Carbohydrates can also be responsible for this self-recognition. And self-recognition is an important part of our immune system. Your immune system recognizes which cells are yours and which are not. And if you have cells in your body that are not yours, your immune system typically will attack them. Generally, this is a good thing. Occasionally, people get autoimmune diseases, and that doesn't mean that you're immune to cars, but it does mean that you become sensitive to your own cells, and your immune system starts to destroy them, and it recognizes cells that it thinks should be destroyed. Cholesterol is in the membrane. The cholesterol tends to make the membrane a little more fluid. It stabilizes the membrane, and it helps keep the membrane sort of liquid even at low temperatures. Although in human cells, low temperatures are usually not a problem. Okay, there are short-chain sugars attached to some of the proteins and some of the lipids within the membrane. And these are called glycoproteins or glycolipids. And they also are important in recognizing which cells are yours and which are not. They are the molecules that determine blood types. You're probably aware there are two major types or sets of typing for blood. There's the ABO system and the RH system. If you have the wrong kind of blood cells in your body, your immune system will attack them generally and destroy them. And not only do they attack and destroy blood cells, but if you have cells from some other person, let's say you have organ transplant, these are the molecules that result or cause your immune system to reject a transplant. That's not good news typically, and in fact there are things that can be done to diminish the sensitivity of your immune system to foreign cells, and that can allow a person to have a transplant without rejection. That's good news. Bad news is typically, though, if you reduce sensitivity and your immune system is not working as well, then you are subject to having diseases and things of that nature that you might normally just be able to deal with easily. Excuse me, some of the glycoproteins are receptors for hormones. They're receptor sites. Remember, hormones are ways of turning cells on and cells off. Sort of the way we can do with nerve impulses. So, when we look at organelles, and this part, the next few slides are actually included in that supplement just to sort of get you going there. But when we look at organelles, we find that there are basically two categories of organelles. We can classify them as organelles that have membranes. These are membranous organelles. and organelles that don't have membranes. Those are non-membranous organelles. So what we'll start is a list of the organelles that have membranes. You want to be copying these down. And here's one of the beauties of the Camtasia. Well, actually, there are a couple. One is that you can listen to me multiple times every day and think about it. I mean, how much better could it be than that, right? But if you miss something in class, you can listen to the Camtasia and sort of fill in the things that you miss. So it should be really advantageous for you. We have the nucleus. We already talked about the nucleus a little bit, and in that supplemental lecture, there's more discussion about it. Endoplasmic reticulum. Remember, the nucleus is the control center for the cell activity and reproduction. Endoplasmic reticulum is sort of like a manufacturing plant where proteins and lipids are produced. There are two kinds of endoplasmic reticulum. There's rough endoplasmic reticulum, and rough actually, it turns out, is one of Pansy the Answer Dog's favorite words. She likes to say rough a lot. In fact, you may even hear her say that during one of these lectures. Now, the other kind is the smooth endoplasmic reticulum. This tends to be where lipids are formed and where glycogen, a polysaccharide, can be formed or broken down for the storage or release of glucose, and glucose ultimately is used for energy release. The Golgi apparatus, or Golgi complex, I like to think of the Golgi complex as sort of like the post office or the Federal Express office, where things that were produced in the endoplasmic reticulum can be packaged either for export or for use within the cell at some other location. All of these have membranes. Okay? We have lysosomes. And lysosomes are vesicles, little balloons, if you will, within the cell that contain enzymes that digest materials. They could digest dead cells or things of that nature, or even in white blood cells, lysosomes can be used to digest foreign organisms. Peroxisomes. Peroxisomes are sort of like lysosomes. They're little bubbles, except they're smaller generally, and they have chemicals in them that allow them to detoxify chemicals within your body. We find these especially in the liver and the kidneys. We have vacuoles. So vacuoles are sort of bubbles too, but they're usually formed at the surface of the cell, where the cell engulfs some particle or other cell or something of that sort, or it could be a molecule. Mitochondria. Mitochondria is plural. Mitochondrion is singular. And the mitochondria are sometimes referred to as the powerhouse of the cell. Mitochondria, we'll talk more about those. There's very good evidence that they actually were not originally part of our cells, that they were an invading organism. And the mitochondria are really good at metabolizing molecules, glucose, proteins, lipid, or fragments of these molecules, and then releasing energy and trapping the energy as ATP, adenosine triphosphate. They're better at doing it than the rest of our cells, so having mitochondria is actually a big deal. Then we have non-membranous organelles, and one of them is the nucleolus. We mentioned the nucleolus already. Nucleolus excuse me, is an area within the nucleus, and it's where ribosomal RNA is formed. We have the ribosomes. Ribosomes are the workers in the factory. Again, all these are organelles that do not have membranes around them. So we'll see that in that supplemental lecture more about ribosomes. The centrosome is going to be organizing the microtubules within the cell, and it has the centrioles, which are sort of microtubules that come in handy for a number of different activities. We have cilia. Cilia is plural, cilium is singular. Cilia usually occur in as multiple organelles as opposed to a single one on a cell. And these are important for moving materials adjacent to the cell in human beings. In single-cell organisms, they're used to move the cell itself. We have flagella. Flagella are responsible for moving cells, and specifically the spermatozoon uses flagella to move through the accessory organs of the female. And then we have the cytoskeleton. Cytoskeleton I mentioned already. This is fibers within the cell that give the cell form, anchored organelles within the cell, real important in stabilizing the cell and the organelles. And she's getting really exciting at this point. So centrioles, we'll talk a little bit about those. Centrioles are short cylinders. Typically they're found actually in a bunch of places. One is associated with the centrosome. And in the centrosome there are two that are perpendicular to each other. These end up being really important during mitosis, and we'll talk more about mitosis near the end of this lecture. Also, there's a centriole in the center of the cilium, and that centriole is attached to the basal body, also with the flagellum, and this directs the formation of these organelles. So here is a centriole. In the flagellum and in the cilia there are two central microtubules as well. This is just one without those. Okay. So it tends to be 9 sets, none in the center. In the cilia and the flagella, they have the 9 sets on the outside plus 2 in the middle, these microtubules. And this is associated with movement. This is the function. Centrioles are important for that. The exact way it works is not well understood. Some people have abnormal cilia or abnormal flagella. Well, generally this is a genetic defect, and if you have abnormal cilia, you also have abnormal flagella. And there is a problem in these people in that these organelles don't work as they should, and so people with this mistake in their DNA tend to have problems clearing their respiratory passages with mucus, and they end up with respiratory diseases. And because the flagella aren't working, or the cilia and the accessory organs of the person, they also have sterility issues. Okay. Now, as I mentioned, the plasma membrane is selectively permeable, and what that means is that some things can go and come out of the cell, go into the cell, and others don't. And this is both going to include molecules and ions. If we have passive exchange, the material moves in and out of the cell without any energy being used to do the movement. If we have active exchange, energy is used, and the amount of energy can be really quite substantial. It can go up as high as 40% of the cell's energy budget being used to move materials through the cell membrane. Okay, so we're going to talk a little bit about simple diffusion. Simple diffusion is good news and bad news. I always tell my anatomy students that when there's good news, there's bad news. And sometimes when there's bad news, there's no good news. This is a case where there is good news, but there is bad news. And the bad news is that this movement is random, which means that they're not directed. They go all over the place, and they may not go where you really need them to go. There is no organization of the movement. That's the bad news. The good news is it's free. The reason these things move about randomly is as they move, they run into two objects, either other molecules or maybe a cell membrane or whatever, and when they collide, they bounce off and go in a new direction, and usually a new speed as well. So the overall consequence of diffusion is that the materials move and end up being equally distributed throughout the volume that they are diffusing within. Net movement is from high to low. That is, materials tend to go from a high concentration to a low concentration. And this is sometimes referred to as moving down a concentration gradient. We haven't talked about gradients before, so a gradient is similar to a hill. That would be a physical gradient. And if you had a car at the top of a hill and then a road going down the hill and a parking lot at the bottom of the hill, if you have the car at the top of the hill and shut the engine off and take it out of gear and release the brake, it will roll down the hill to the bottom. So it goes from high to low. If you're at the bottom of the hill and you release the brake and shut the car off and take it out of gear, it will not roll up the hill. So that may be a good functional analogy, because it can kind of help you remember that things tend to go from high places to low places spontaneously. Here is the path of a particle that might be moving around on a random path. So we see it going over here, it comes along and it hits something and it bounces off, and it goes to here and it hits something else, it bounces off, it goes over here and it bounces and bounces and bounces, and it goes zigzagging all over the place. In this case, it went a long way before it hit something. Here, not so much, kind of a short path, and eventually ends up here. Now, if you straighten this string out, you could see that it would be a whole lot easier if it just went from here to here, but that's not how diffusion goes. It's always random. There are a couple of really important principles that emerge that affect diffusion. You should be aware of these. You might even see these things covered on a quiz or something of that nature. Oh my God. The greater the differences in concentration of a substance, the higher the net rate of diffusion. Net rate just means that they're moving in one direction overall more quickly. If they're moving in two directions and the amount of movement in both directions is equal, there is no net diffusion. But if you have a concentration gradient, like a hill, it goes from high to low, and the greater the difference between the concentrations, the faster the diffusion rate. Smaller molecules are capable of faster diffusion than big molecules. This is pretty simple. The reason these molecules are moving is because the energy they have. And so if you have two vehicles, let's say, we'll give you an analogy to help you with this. If you have two vehicles and they both have exactly the same engines and one of them weighs 10 times as much as the other one, the one that's light will go a whole lot faster than the one that's heavy, right? It's the same thing with molecules. At a given temperature, all the molecules have the same average amount of energy. So that means that the small molecules move faster and the big molecules move more slowly. When we talk about small and large, we're not talking about size as much as we're talking about mass. Another general principle is that the shorter these things have to go, the faster the diffusion occurs overall, and that's simply because it's random. They don't have as very far to go, so they get there more easily. This turns out to be a really big deal because where we have diffusion would be, for example, in the lungs or with the capillary beds, and capillaries have to be really close to whatever they're exchanging with in order to be effective. The further the capillary is from the cell, the longer it takes things to get there, and in fact the cell might even die while it's waiting. The fewer physical restrictions, the higher the rate of diffusion. This is pretty simple. You got these molecules bouncing around, ions bouncing around randomly. If there are barriers that inhibit their movement in any given direction, they move that way more slowly, and the more barriers, the slower the rate of diffusion. The fewer the barriers, the faster the rate of diffusion. Pansy's eating some candy, I think. Some more things. Higher temperatures, higher rate of diffusion. This is pretty simple because you want to understand when you measure the temperature, what you're really measuring is the amount of kinetic energy the molecules have. That's what temperature measures. If they have more kinetic energy, they move faster. It's that simple. It's sort of like putting the accelerator down in your car. The more energy you have coming out of the engine, the faster the car goes. Viscosity. We haven't talked about viscosity before, but viscosity is a measure of the resistance to flow, and things that aren't very viscous allow for faster diffusion. I'll give you a different measure of viscosity, an example. Honey is viscous. Water is not as viscous. So things would diffuse more quickly through something like water than they would through something like honey. Just to give you an idea about how fast these molecules are going, I think this is sort of an impressive statistic. Water molecules at body temperature have an average speed of about 1,500 miles an hour. This is pretty incredible. So I'm going to ask Pansy a question here, but you guys can be thinking about this too. She's always good at coming up with answers, you know. So if all of the water in your body is moving at 1,500 miles an hour on the average, and about 60% of your body mass is water, how come you don't fly in one direction at 1,500 miles an hour? Because all the water is going that fast. And the answer is right, Pansy. She can even do that while she's eating a piece of candy. Not all the water molecules are going in the same direction. They're going in random directions, so they sort of cancel each other out in terms of the effect on your body moving around. But next time you bump into somebody, you could claim that the reason you bumped into them was that most of your water molecules were moving towards them, and it made you do that, and you're sorry. So, once the concentrations balance out and throughout a volume, we have what's called equilibrium, diffusion equilibrium. And this doesn't mean that the molecules aren't moving anymore. It just means that there's really no net change in concentration throughout the volume. Okay, so that would be a point where we would have the net flux would be 0. Again, I want to stress that net diffusion goes from high concentration to a low concentration, and it's only effective over short distances. So I've got a video that's sort of a kind of a cool thing to watch. And Pansy likes watching this especially. It actually makes her go into a coma. So here we go. What we're going to do is I'm going to try to, oh boy, I'm going to try to get all the red spheres on the same, oh boy, same side of the membrane. The membrane will be in the middle here. There we go. We're almost getting it. I've got to wait now. And we'll let those, for argument's sake, we'll pretend that the red spheres are, almost. We'll pretend that the red spheres are oxygen molecules, and we'll pretend that the blood, okay. We'll pretend that the blood is on the left-hand side of your screen with oxygen-rich blood, and we'll pretend that you have a muscle cell on the right side of your screen with oxygen-deficient blood, and we see that the oxygen molecules don't all just run over to the muscle cell. They continue to exist in both the blood and the muscle cell, but we see that overall, this is going to make me alive. There we go. We're almost balanced now. Yeah, we are balanced now, right? We've got 5 on each side. Now, it took a while for that to happen. Could some of the oxygen molecules in the muscle cell go back into the blood? Yep, one just did. Would you ever expect, once you get an equilibrium of concentration, would you expect them all to leave the blood and go into the muscle cell, or all of them leave the muscle cell and go back to the blood? Nope, they kind of balance each other out. So you see, even though they're coming and going, the concentrations are staying pretty much the same. I know many of you would like me just to continue to play this game for the rest of this Camtasia, but we have to move on. So Pansy's rolling her eyes. She's not very happy about that. Here's an example of the effect of diffusion. So this came from a physiology book that I used a long time ago. You could get 10% of the blood glucose to diffuse across a capillary, which is about the size of a red blood cell, in about 3.5 seconds. To get it to move 10 centimeters, which is about 4 inches, it would take 11 years. You don't have 11 years to wait for your glucose to get to your cells. So what this means is you would expect that all cells would be really close to capillaries. I think it's probably better that she chew a piece of candy than bark all through the whole Camtasia recording. So we're just going to have to kind of deal with it. I hope it doesn't upset you too much. Okay, also we have a portion of the membrane that's relatively solid, made up of the phospholipids without pores. And generally the kinds of molecules that diffuse through the solid portion of the membrane will be lipid-soluble molecules. And so I'm guessing that some of you could figure this out, and even Pansy probably has an opinion on this. So why would lipid-soluble molecules go through the solid portion of the membrane more easily than materials that are not lipid-soluble? Right. The lipid portion of the membrane in the middle would act as a barrier to polar materials, and lipid materials can go right through that. So the lipid nature of the membrane is responsible for this. Nice thing about molecules that diffuse through the solid portion of the membrane is that you don't have to give them any extra energy to get them to go through. However, I should note that the membrane does slow things down a little bit. The good news for us is that even though it does slow things down, it's not very thick, so it doesn't take very long for these molecules to go through. So here would be an example of a molecule starting on the left-hand side of your screen and diffusing through the solid portion of the membrane to the right-hand side of your screen. It could be leaving the cell or entering the cell. Okay, and examples of molecules that are lipid-soluble and relatively small are carbon dioxide, oxygen, and alcohol. So these things can go through membranes relatively quickly. That's important because carbon dioxide and oxygen are the respiratory gases, and we are producing carbon dioxide in our cells. That's a waste product that we have to get rid of. And the oxygen we need to get into the cells so that the cells can break down organic materials. Osmosis. People use osmosis incorrectly quite frequently. They don't understand really what it means. Osmosis is the net movement of water due to the concentration of water molecules. Notice that I made water molecules here capital. It's not the concentration of other things, it's the concentration of the water that's really important. And it's always passive. So if on one side of the membrane you have a lot of water, and on the other side you don't have very much, the water will go from where it's concentrated to where it's not concentrated. Now, for osmosis to be most effective, the solute has to not go through the membrane, because if the solute is going through the membrane, it's going to affect the water concentration as that's being moved. So this is where the selectively permeable membrane comes in kind of handy. So in osmosis, water is going to go from high to low. So let's look at this diagram that I brought in here. On this side of the membrane, it's all water molecules, right? That's all there is. So it's got a high concentration. On this side of the membrane, there's not as much water because we have some big protein molecules. So which way would you expect the water to go, from top to bottom or bottom to top? By the way, it's actually going to go in both directions, but the net movement is going to go from from top to bottom, because the top has the higher concentration. Very good. She's really good at this. Okay, so the water is going to move toward the bottom portion of that diagram. If the bottom portion is inside the cell will swell, and if it swells too much, it may actually rupture the membrane. If it's outside the cell that has less water, then the cell shrinks. Okay, so here's kind of an interesting thing. If you apply pressure to the side of the membrane, or the material on the side of the membrane with low concentration, that pressure basically compresses the material and effectively raises the concentration. So I've got a slide to show you this. If you get that pressure high enough, it will stop net diffusion, and that amount of pressure that you're applying is called the osmotic pressure. When we get to blood, we'll be talking about osmotic pressure. So here we're going to have some gas cations in a cylinder with a piston. Now, we understand that this is not a liquid, but gases are a really good way to describe this, because I think you can kind of sense what's going on here. So let's say we have this cylinder, and we have 5 gas molecules. They're cations. You can see they're cations, right? Pansy loves cats. And if we put pressure on the piston and push it downward, these cations become more concentrated, right? So the same thing happens in fluids. You don't see a big change in volume, but by exerting a pressure, you do an external pressure to a volume, you increase the concentration within that. Okay. So, tonicity. This is the degree to which the concentration of solute causes osmosis to occur. So, remember, osmosis is based on the water concentration. But usually when we talk about solutions, we don't talk about the water concentration. We talk about the concentration of the material in the water, the solute. And so there are 3 kinds of solutions. There are isotonic solutions. Isotonic solutions have the same number of particles inside and outside. We have of a cell, for example, or you could have a solution that isn't outside or inside of a cell. You could just have a solution that's isotonic. And some of you probably even consume gin in isotonic, or vodka in isotonic, right? Those are good drinks. We have hypotonic, which means that there is more water and less solute. And we have hypertonic, which means there's more solute and less water. So we'll consider each of these. We'll start with an isotonic solution. An isotonic solution has the same number of non-penetrating particles as the cell. And we're not going to really get into molarity, but this is just a measure of concentration. A 55.2 molar solution is isotonic. If you have an isotonic solution, and this is with respect to water, if you have an isotonic solution, cell volume does not change. Pure water is 55.5 molar. That means there isn't anything there except water. Now, if we have intravenous solutions in a hospital, typically those solutions are going to be isotonic, and you want that on purpose. If you have a solution and it is not isotonic, your cells are either going to swell up or shrink and shrivel. Neither of those are desirous outcomes. Okay, we have hypotonic solution. That means that you don't have very much of the solute and you have a whole lot of water. Okay, and that means that the cell will swell, and that's because there's more water outside than there is inside the cell. The water moves from high to low, goes into the cell, cell swells up, and if it bursts, it's called lysis. Some of you may have heard of hemolysis. That's when a red blood cell bursts. So if you were to inject pure water into somebody's circulatory system, their red blood cells would rupture and a person would become anemic real quickly. That would be undesirable. Hypertonic solutions. Hypertonic solutions have a lot of solute and not as much water. And that means that the high concentration of the water is inside the cell, low concentration outside the cell. The water goes from high to low, so it moves from inside to outside, and the cell shrinks. Okay. In red blood cells, this happens sometimes when people are making slides of red blood cells, and it's called crenation. Actually, the cells often look kind of bumpy when you see a crenated cell. Filtration. Filtration is an entirely separate process. It tends to happen in capillaries, and the plasma is going to, or some of the plasma is going to leave the capillary and go into the interstitial fluid. The things that won't leave are the things that don't fit through the pores of the capillaries. Capillaries tend to have a lot of pores. They also tend to have a little bit higher hydrostatic pressure than they have outside the capillaries. Hydrostatic pressure just is when you measure blood pressure, that's hydrostatic pressure. So the pressure inside the capillary causes things to leave, and basically the things that don't leave are the things that really are big and don't fit through the pores. The cells, the plasma proteins, they tend to stay inside. So this is filtration of the blood. We use that actually in the kidneys to get materials out of the blood, and then we have selective reabsorption so that some things are lost from the body and some things are brought back in and retained. Here's the analogy of the coffee maker. I think it went too fast. I didn't set that up properly. So in the analogy of a coffee maker, water goes through the percolator, it percolates, right? And the things that leave the coffee grounds are going to be the flavor and the color and the caffeine and water, and the grounds, the large particles, are left behind. And the reason they're left behind is because they don't fit through the pores. So it's the same thing in your blood. Transport by carrier molecules through the cell membrane. This is a big deal. Most things that enter the cell come through by way of carrier molecules, and they can't get in any other way because they're too big or because they're not lipid soluble. So too big would be like having a doorway with a piano on one side, and you want to get the piano through the doorway, but it won't fit because it's too large. So it doesn't come in. There are ways to get it in. Like the example I used before, if you're going between floors, you could use an elevator. Or they're not lipid soluble. So if you have something that is polar compound and it's big, it doesn't fit through the pores, it won't go through lipid materials. Even if it's small, it won't go through lipid materials because likes dissolve likes, and things that are different tend not to dissolve very well. And then we have facilitated diffusion. Facilitated diffusion has the word diffusion in it, so you would expect that things are going to move from a high concentration to a low. That's true, but in facilitated diffusion or facilitated transport, you have to have a carrier molecule involved because the material won't diffuse otherwise. And this is going to be really important for getting glucose into the cells and amino acids into the cells. You might think, well, if it's diffusion going from high to low, don't they diffuse back out? In theory they would, but typically when glucose goes into a cell, the cell modifies the glucose so that it can't leave, and so it's sort of trapped. And the amino acids get used up, so they don't leave either. Okay, so carrier molecules are going to accept the substance that's going to be transported. We can take glucose as an example, and they carry it through the membrane. And so you might wonder, how can they do that? Generally, when the glucose attaches to the carrier molecule changes shape, and it because of that change in shape, it moves through the membrane. Okay. As I mentioned before, because it is diffusion, things go from a high concentration to a low concentration. And it does allow for two-way flow, so there's a possibility for things to come back. But as I mentioned, the cells are pretty clever about that, so when glucose moves into the cell modifies the glucose so that it can't escape. Oh my God, that candy's going to go away pretty soon, I hope. Okay, there is going to be a little bit of energy required for facilitated diffusion because you need to have thermal agitation. You don't really need to worry about that. It doesn't require a lot of energy, but it does usually require a little bit. Okay. The rate of facilitated diffusion is going to be determined by several things. One is the magnitude of the concentration gradient. So the bigger the gradient, the faster the stuff diffuses. One is going to be the attributes of the carrier molecule. So if the if the carrier molecule has a certain shape, that will affect how quickly the material can go through. And the number of carrier molecules in the membrane will affect it. So if you have more carrier molecules, you can get more material in. The analogy I like to use here is the carrier molecule concept is a little bit like what happens at a restaurant. So you go to a restaurant that has a person who helps you be seated, and you have to wait until that person's ready to take you to your seat. One way that you can seat more people would be to have more people come out and accompany the guests to their seats. If you only have one person, only one carrier molecule, things are going to go more slowly than if you have a bunch. Okay. This is a type of mediated transport, mediated by a carrier protein. Another thing is that once the carrier gets saturated with how many molecules it can move, the rate levels out. It really doesn't matter how many people are standing in line outside the restaurant. The waitstaff can only bring a finite number of people to their seat in any given time. You need to have more people making the movement if you want to get faster transport. Okay. Active transport. Active transport is a little bit like facilitated diffusion. It involves a carrier molecule, but the big difference here is that molecules or ions can be pumped through a membrane regardless of what the concentration is. So in facilitated diffusion, the net movement is from high to low. In active transport, doesn't matter what the concentrations are. They tend to go in one way. Generally, the carrier molecule is going to be a protein, and a lot of energy is required in order to get this movement to go. This is sort of like pushing a car up a hill. It takes a lot of energy to do that. Can you do it? Sure, you can do it, but it does take a lot of energy. Okay. The energy source is going to be ATP. We've talked about that before. ATP basically provides energy for three activities. One is moving things through the membrane, another is chemical synthesis, and another is mechanical activity like muscle contractions or things like that. What it appears to be is that the energy is probably attached to a phosphate ion and to the carrier molecule. You use that energy to make that attachment, and that attachment of the phosphate brings the material through. Again, this is going to be limited to a maximum rate, which is going to be determined by how many carrier molecules there are and how much energy is available. So here we have active transport. Active transport, you can see, is sort of like using the elevator, right? But it's very specific on what it moves. The shape of the molecule determines what can be moved and what won't be moved. And in active transport, the material only goes in one direction, whereas in facilitated diffusion, it has the potential to go both ways. Okay. I mentioned the phosphate groups are probably become associated with the carrier molecule, and that allows for the motion to occur. So that phosphate group changes the carrier's shape, and that takes it through the membrane. So one of the important active transport mechanisms that we have in cells is the sodium-potassium pump. This is hugely important in nerves and in muscles. Okay, and the same carrier can move the sodium and the potassium through the membrane, except it moves them in different directions. What happens is the sodium gets pumped out of the cell, and the potassium gets pumped into the cell. But it's a little bit asymmetrical. So it's better at moving sodium than it is at moving potassium. So about 3 sodium ions move for every 2 potassium ions that get pumped in. And it takes 1 ATP molecule for each cycle of moving a sodium out and a potassium in. Okay. Because it's asymmetrical, you end up with a charge building up on the outside of the cell, because more positive charges are leaving than coming in. And so the outside of the cell becomes positive and the inside of the cell becomes negative. Now, at the same time that the sodium and potassium are being pumped, they also can diffuse through pores. And there are different factors affecting the two ions. So we'll take a look at that in a moment. Okay. As I mentioned, a lot of that energy can be used in these active transport, and in the sodium-potassium pump, between 10 and 40% of the cell's energy is expended on the pump. So where does the energy come from? Pansy knows the answer to this. Right, ATP. She can talk while she chews candy. She's unbelievable. Okay. As I mentioned, this mechanism is really important in muscle cells and in neurons, and it affects the activity of both. We're going to talk more about that when we get to topics 7 and 8. So, in addition to moving these positive ions, negative ions also move. Negative ions are going to move through the channels to follow the electrochemical gradient. And so, because there are more positive charges moving out of the cell than moving inward, the chloride diffuses out of the cell to follow those positive charges. In people with cystic fibrosis, this mechanism doesn't work properly, and that ends up being really problematic for them. Another kind of transport that we have is endocytosis and exocytosis. Sometimes these are referred to as vesicular transports because vesicles are involved. And in endocytosis, engulfment of the material actually occurs. The cell kind of surrounds what is going to be brought into the cell, plus some of the fluid outside the cell gets brought in as well. So here is a case of a cell beginning endocytosis, or pinocytosis is a subdivision of endocytosis. This actually means the cells drinking. So the process begins when this particle, in this case a protein, is outside the cell completely, in the interstitial fluid. And what happens is that, the cell starts to surround this particle, okay, and this, what's starting to form here is a vesicle. Here the vesicle is almost completely formed, right? There's the protein and interstitial fluid outside. And then eventually we have a vesicle with a protein, and this fluid in here is the interstitial fluid or extracellular fluid. It got taken in with the protein. Phagocytosis is a specific type of endocytosis. Some of you may have heard of phagocytosis before. Phagocytosis is when a cell surrounds some object outside of itself. White blood cells do this when they ingest foreign cells or particles. And typically in a white blood cell, once this material is ingested, the cell tries to digest the particle, break it down, and it uses the lysosomes to do that. Infants use pinocytosis actually to get the antibodies from the mother's blood that they have consumed. It's a type of endocytosis. Exocytosis is sort of the reverse of pinocytosis. And here we have materials that are inside the cell in vesicles being released to the outside typically. The things that could be secreted would be proteins, for example, insulin from the pancreas, or enzymes like salivary amylase with the saliva. So here we have a vesicle inside of the cell, okay? Intracellular fluid here, and the extracellular fluid is out there, or interstitial fluid. Here the vesicle has come in contact with the membrane and is starting to rupture. Here the ejection of the particle is almost complete. And then here the ejection is complete. At this point, this particle may diffuse into the blood or lymph, or may be in the region for a while. Okay. So the next thing we want to talk about is the cell cycle. Cell cycle involves division of the cell, and then all of the things that the cell does after it's divided. So we can separate the cell cycle into a bunch of different periods of time that are defined with respect to the cellular activity. The cell division is controlled by chemicals. Some inhibit the division, others stimulate it. And apoptosis or ptosis is when a cell undergoes abnormal meiotic activity, and typically apoptosis leads to the death of the cell. I think I must have had too many beverages at lunch. Okay, so some cells become specialized after a certain number of cell cycles, and they stop dividing. Examples of those would be muscle cells and nerve cells. They either rarely divide or never divide once they've gone through a predetermined number of cycles. Okay. Other cells stay in sort of a primordial, if you will, state, a very undefined state or not specialized state, and these are essentially immature cells. It's like stem cells, and they continue to undergo cell cycles for an indefinite amount of time. These stem cells then are really important for recovery from damage to cells or tissues or organs. So when you get an injury, the way you develop new cells within your tissues is through the cell cycle. It also is responsible for growth as an individual goes from a single cell to a multicellular organism. Okay, there are a bunch of stages within the cell cycle. We have interphase. Interphase, when I was in college, we sort of said, oh yeah, well the interphase is really when cells do everything else, and it was sort of looked at as really not being an important part of the cell cycle. But really this is the most important part of the cell cycle, because this is when cells do their thing. This is when they do their specialized functions. If they're dividing, they're pretty much restricted on what they can do. During the interphase, chromosomes are doubled and the cell prepares to divide. So here we have some centrosomes. You'll notice there are two of them, a nucleus within the cell. Okay. There are several parts of interphase. We have G1 phase, which stands for the growth of the cell. The G stands for growth. This is before the DNA is replicated, and the cell is returning to a condition that will allow it to go through mitosis eventually. The cell grows, and the numbers of organelles within the cell doubles. So cell organelles are dynamic in terms of their abundance. That's kind of an important thing to bear in mind. Okay. Proteins are synthesized during the G1 phase. So if you have a secretory cell in a gland, let's say, they might be forming the enzymes that that gland releases, or maybe hormones that the gland releases, if it's a protein hormone. The DNA prepares for synthesis of the various compounds the cell could produce, and the normal specialized functions occur. So this is when muscle cells would contract, or nerve cells conduct impulses, or glandular cells secrete substances. There's an S phase within interphase, comes after the G1 phase, and this is when things are synthesized. The DNA is replicated. That's a synthesis process, and we start out with a 23 pairs of chromosomes, from 23 from each parent, and these chromosomes initially are just one chromatid, just one piece, one strand. At the end of this synthesis, the DNA synthesis, there'll be two chromatids. These are called sister chromatids, and they should be identical in terms of their composition, at least if everything goes normally. Then there's a G2 phase. In the G2 phase, the proteins associated with the microtubules are synthesized. The centrioles are duplicated, replicated, and we'll talk more about those. And so now let's talk a little bit about the duplication or replication of the DNA. When the DNA in the chromatids is duplicated, it is essential that the DNA be made into an exact copy. You don't want to have a lot of variation with the DNA, and so the duplication results in sister chromatids that have pretty much exactly the same information. Now, is it possible for mistakes to occur? Yes, it is. And when those mistakes occur, it usually is sort of unfortunate, but that does happen. So each of the original parental strands of DNA acts as a template, and you get a new daughter strand that is exactly the same. The DNA molecules are held together by hydrogen bonds. We talked about those back in the chemistry lecture. Those hydrogen bonds break and the strands unwind. A good way of thinking of this is sort of like a zipper, I think. So if you think of a zipper, it's got two sides, and think of the zipper being held together by hydrogen bonds, and when the zipper unzips, those hydrogen bonds would be broken. Okay. There are nucleotides within the nucleus, and the nucleotides are adenine, thymine, cytosine, and guanine, and they are like raw materials that can be incorporated into the new DNA strands. And so there are these purines and pyrimidines. They're joined by an enzyme called DNA polymerase, and you can tell it's an enzyme because it has ASE at the end of it, its name, and that's going to form the new daughter strands. So at the conclusion of the replication of the DNA, you have two identical DNA molecules present. And that means that the new chromosome is going to have two chromatids with exactly the same information. Again, mistakes do occur, but most of the time things go normally. The DNA serves as a template for the synthesis of RNA. So it has a code, if you will, defined by the exact sequence of the nucleotides within the DNA. And there are going to be two steps in protein synthesis. First, we have transcription. And transcription occurs when messenger RNA forms from the original DNA molecules. And this messenger RNA will have a code. It's called a codon, or direction for a group of amino acids being joined together. Translation is when the messenger RNA specifies the order for those amino acids, and you end up with a protein molecule. I think the candy has all been consumed. Good thing. So there are going to be three bases associated with the code. This is a codon, and that's going to be within the messenger RNA molecule. So you can think of groups of three, each of which specifies a specific amino acid. The gene then is going to be the set of triplicate codes for one complete protein or polypeptide. And that gene then should give the instructions for the cell on how to make whatever that protein is. Okay, so in order for this transcription to occur, the DNA unwinds much like it does when it replicates itself. And the parental code acts as a template for the formation of the messenger RNA strand. The nucleotides that form the RNA are also in the nucleus, and they are brought together again by another enzyme, RNA polymerase, to make the RNA molecule. So, yeah, is that? It's like I'm psychic or something. There are three kinds of RNA molecules. There's mRNA, messenger RNA, that has the code for the exact sequence of the amino acids in a protein. There's rRNA. rRNA is ribosomal RNA, and it makes up part of the ribosomes. And then there's tRNA, the transfer RNA, and that's like a gopher that picks up the appropriate amino acids, brings it to the site of the protein synthesis, and releases it. So I've sort of been talking about this in abstract terms, but here is what we have. So let's say that we have a DNA molecule in it, and it unzips. On the left-hand side here, we have the code for two amino acids. The first one is thymine, adenine, cytosine. Second one is adenine, adenine, adenine. Then we have a non-code strand of the DNA that matches with this. And these, the amino acids specified here are methionine and phenylalanine. These are not random, right? So A always goes with T and C always goes with G. Okay. And so you can kind of think of this non-code piece of the DNA as keeping this one protected and just kind of being bound to it under normal circumstances, and it unbinds when it's time to form a new molecule. So here we have the messenger RNA being formed. Okay. And so adenine goes with thymine, and in the RNA, instead of thymine, it has uracil going with adenine. Guanine goes with cytosine. So here we have AUG, UUU. This would be the code that would be used to get the amino acids for the protein synthesis. Okay, so this is the sense strand of the DNA, and the other strand is not used in the protein synthesis. As I said, it's sort of, you can think of it as sort of like a sheath that a knife goes into. Oops, sorry. Okay. So the messenger RNA then determines the amino acid sequence of the protein. As I mentioned, there are three kinds of RNA. They're all needed to form proteins, and the process of forming a protein is called translation. So transcription was the formation of the messenger RNA from the DNA, and translation is taking the messenger RNA and actually forming the protein. So if you think of the physical arrangement here, the messenger RNA is sort of like the filling of a sandwich, and the ribosomes would be the bread. There's going to be one on either side of the messenger RNA. And then the transfer RNA is going to be a gopher. It goes out and it brings the amino acids into the ribosomes. Okay. So there is protein and ribosomal RNA making up the ribosomes. Those, remember, I hope you did the lecture and Camtasia for the cellular organelles. The ribosomes are those little workers that actually are physically involved in forming the protein. There's going to be a specific transfer RNA for each of the amino acids. And in normal protein synthesis, there are 20 amino acids that get used. So what happens is the transfer RNA attaches at one end to the amino acid, and then the other end of the transfer RNA has an anti-codon 3-base sequence that matches the messenger RNA. And when they line up, it brings that, releases that amino acid for the protein synthesis. Okay. So the anticodon of the transfer RNA attaches to the codon from the messenger RNA. The amino acid is released and incorporated into the protein that's being synthesized. This is pretty cool. Actually, the fact that it all came about is pretty remarkable. All right, so we have a couple of stages within mitosis. We have nuclear division. We have, and that's called mitosis. We have the cytoplasmic division, that's cytokinesis. And when these two combine, we end up with two new cells, daughter cells. They're usually referred to as daughter cells, and they should be identical in terms of their genetic material. You remember that there's a second growth phase during interphase, and that's when the centrioles get duplicated. So there are going to be two of them within the cell that's prepared to go into prophase. Prophase is the first part of the dividing activity for cells, and the two centrioles move to opposite ends of the nucleus. It is possible that they get pushed apart, or they may get pulled apart. It's not really clear at this point exactly which of those two processes is going on. You might think, well, why don't they know? All of this is pretty small and dynamic, and so it's not like you can just say, oh yeah, there it is. It's a little difficult to discern exactly what is happening. Also in prophase, the chromosomes condense. Chromatin is the material in the nucleus that really eventually becomes the chromosomes. It sort of coils on itself. Also, the nuclear membrane breaks down. This is sort of essential because you're going to have to form two new nuclei. Okay. Spindle fibers, which are tubules attached to the centromeres of the chromosome. Centromere of the chromosome is where the two chromatids attach. So you've probably seen pictures of chromosomes. They sort of look like an X, and the waist of the X, where the two pieces come together, that's the centromere. And those start to pull toward the centrioles. So the spindle of these fibers is complete by the end of prophase, and we have two poles. These are formed by the centrosomes. It's going to be one at each pole, and there are going to be asters radiating from those poles with the fibers. So the spindle fibers exist between the poles. We're going to have some pictures here shortly. The centrosome is sort of like the hub of a wheel, and then the fibers, the asters, are like the spokes of the wheel. They radiate outwards from the centrosome. So here they are. So there's the centromere right there. I think you can see this looks kind of like a narrow X. Here's also kind of an X with the centromere right in the middle. Okay. So it's the centrosome, the spindle fibers attached to the centromere of the chromosome. They're the sister chromatids forming a chromosome, and the nuclear membrane is broken down. Then we have metaphase. And metaphase, by the time metaphase comes around, the nuclear membrane is completely gone. So it's no longer containing the DNA. The chromosomes line up along the middle of the cell. This is sometimes called the equatorial plane. So here is the equatorial plane. And here you have a chromosome, another one, and I think you can see they sort of look like really skinny X's. They join at the centromere, right? And those spindle fibers, those spindle fibers are going to start pulling the chromosomes apart. So then we have anaphase. If you remember from our zoological roots, ana means to separate, right? So anaphase, things start to separate. We have two chromatids, and these are called chromosomes at this point. So the chromatids are going to move toward the centrioles. One from each pair goes to each end. So here we see, this is anaphase. We have the chromatids pulled apart. The centromere is still there, and they're getting drawn toward the pole. Okay. So Spindle function. These spindle fibers cause the chromosomes to move. And there are two types. One type attaches a centromere and pulls the chromosome toward one pole, and the second type is going to push the chromosome toward the pole. Both of these activities can happen because spindle fibers can undergo two changes. They can get shorter or they can get longer, depending on the activity where they are. So to get longer, they just add these subunits of protein. And to get shorter, they just get deconstructed. And then we have the last phase, telophase. Telophase, the chromosomes have reached the poles, a new nuclear membrane starts to form. Spindle fibers disperse, and the chromosomes, which became sort of coiled and condensed at the beginning of the division, uncoil and uncondense, and they go back to being called chromatin. And then new centrioles form during telophase. So here, there's the new centriole, one at each end. We got the new nuclear membrane is forming, the cleavage furrow, and again, more stuff about telophase. So the nucleoli appear again during telophase. Cleavage furrow appears to be involving actin filaments. Actin filaments are protein filaments that have contractile properties, and this causes the cells to pinch off. So, the importance of mitosis. They're of huge importance. You have to have mitosis from the time you become a fertilized individual cell to the time that you are a multicellular organism and you die of old age and arthritis. We need mitosis in order to grow. We need mitosis to repair tissues that get damaged. Now, there's a second process called meiosis. Meiosis is reduction division, and this is the process that produces gametes or sex cells. And there are two kinds of gametes. There's a male gamete called a spermatozoon and a female gamete called an ovum. So here is a spermatozoon. Up here is where the genetic material is. This is where the mitochondria are that produce energy, the ATP. Well, they don't produce energy, I'm sorry. They produce ATP that's used in the swimming motion. And then we have the flagellum, and the flagellum is responsible for the swimming. Here we have an ovum, and the ovum has an outer membrane called a corona. It has a nucleus, and this is called a polar body. It's basically just garbage. The ovum and the spermatozoa, they couldn't be much more different. Ova are really big, spermatozoa are really small. Spermatozoa swim, ova do not. They're sort of static. But that gets us to the formation of the gametes. In the formation of the gametes, the genetic material gets duplicated once, and then following that, there'll be two cell divisions. So you duplicate things, you get twice as much genetic material as normal, and then divide twice, and you get down to half of the normal genetic material, half of the normal chromosomes. Now, this is not random, so it's an exact half. You get one copy of each of the chromosomes. Some of them came from the mother, some came from the father. And then those gametes would combine and they would form a zygote. And the zygote, this would be the act of fertilization. And the zygote will divide by mitosis until it dies of old age and arthritis, except in the gonads. I didn't mention that, but the meiosis only occurs in the gonads. It doesn't occur elsewhere in the body. So this gets us through quiz 2. I hope that Pansy's chewing of candy didn't drive you too crazy, and I apologize for that, but if I tried to take it away from her, it would have probably resulted in her going on strike and being unwilling to go on. I hope this helps. We'll be going through this material in class. Obviously, you're welcome to use this, though, to kind of fill in any blanks that you leave. When we come to blank places in your PowerPoints, I move right along in the Camtasias because you can pause them when you need to. In the class, I'll give you more time to fill those in. Okay, hope you enjoyed it, and I hope you have a great evening.